Hello, everybody. Welcome to the first mini class of the month. My hope is to do it once a month. Hopefully, I will be able to keep up. And for today, I chose a topic that is extremely important and interesting. And that topic is the brain's emotional development. Okay. So this is not like a master class where I discuss like all of the brain development, but my idea is to pick one or two articles and then sort of talk about the research and what researchers are saying. Okay, so I chose two articles by Dr. Tottenham. She is a neuroscientist who is at Columbia University and specializes in the brain's emotional development. So let's go. So as we know, we are born very immature, right? We're born with 25% of adults' brain size. Like when we are babies, we only have 25% of the brain volume that we're supposed to have later on. Imagine that, right? And so that gives us the first clue about core concepts of brain development. So let's start with a core concept, okay? And one core concept is that the human brain is a neotenous brain, and Dr. Tottenham talks about it. I didn't know even about this word until I read her work. So I was like, what is this? Oh, I need to share with people. So what is neoteny? So neoteny is the delaying or slowing of the physiological development of an organism. So this means that the caregiving environment has a powerful effect on its development. And also because there is the environment has a powerful effect, then it means also there is vulnerable, right, to adversity for a long period of time. So one of the examples is that the prefrontal cortex, right? This part of our brains that we all know develops very later, like ends or finishing develop very late in life. Very late in life, I mean, like as a young adult. So we all know that the prefrontal cortex undergo as very slow synaptic reorganization, okay, until the third decade of life, or 25 years old, more or less. So that's the first clue that the human brain is a neotenous brain, right? So neoteny allows for extended learning opportunities, right, because of brain plasticity, but then it also increases the risk of poor outcomes following early adversity. So another thing that comes because we have a neotenous brain, another thing that follows is that we as adults and as mature adults have remarkable emotion regulation abilities. If we compare ourselves to other species, we are actually very, very good at regulating our emotions. And we also know that childhood development of emotion regulation is very, very, very slow. Okay. So it's like we need it to be this low so that then we have this super capacity when we are adults. I mean super capacity when we compare ourselves to other mammals, okay? So the nature and quality of environmental inputs during these periods of plasticity, in particular those from close relationships like parents, friends, and teachers, in large part determine emotion regulation functioning in adulthood. Right? And this is why I have my open position about sleep training and all, and why I support parenting that is peaceful and that it sets the parent on a track to be the emotion regulation sort of creators of the future emotion regulation of the children. Okay. So as I said, so I'm going to be talking about Nin's paper, right? Dr. Tottenham's paper. So there's this figure I wanted to share with you. And so in this figure from her paper, we see the comparison among species, okay? So at the bottom, we can see how the period of developmental plasticity in green in humans is longer relative to other species. So we have the precocial development, and in blue is the adult characteristics, and on the x-axis you see the developmental time. So it's like basically showing that for precocial species like ducks and horses, they are born and they don't have a lot of developmental plasticity. They're born like doing almost everything already, okay, or almost everything that they need in their adult world. So that's why you see all blue there. Then you have the altricial species, and that's when you start seeing more developmental plasticity in green. And so you see that right on top of the green, you see scaffolding by caregiver. So that's what is our job. We're building these structures for our species, and then the other mammals too, like mice. Like this is why there's a lot of rat research, because we can study scaffolding by caregivers in rats, for example. So here in this figure, she's exemplifying, okay, the development of the amygdala and the medial prefrontal cortex circuitry. Okay, so this slow development of emotion regulation is paralleled by the slow development of the neurobiology that supports it. So we see this in biology, right? So that's why children don't have the emotion regulation capacity that an adult has, because there is slow development of the supporting neurobiology. There is a slow development of the connections, okay, between the medial prefrontal cortex and the amygdala. So then the dotted lines that you see in this figure represent the windows, okay? It's like sensitive periods, okay? There are time windows of development during which the effect of the caregiving environment on brain function is particularly strong, okay? So there are sensitive periods for this. So in this figure, Professor Tottenham shows the developmental window of the amygdala and the medial prefrontal cortex, which circuitry is crucial for emotional development, and its development is slow, very slow. This is why babies cannot learn to self-soothe or going for a high state of stress to a low state of stress by themselves, because they need scaffolding, okay? And other things that I can tell you is that several studies have shown that communication between the medial prefrontal cortex and the amygdala, which supports emotional learning, is qualitatively different in childhood than in adulthood. Okay? We see this in studies. What do we see specifically in adulthood? For example, if we look at the adult brain, increases in medial prefrontal cortex activity are associated with a decrease in the amygdala activity, okay? When, for example, adults are looking at fearful faces, okay? There is an anti-correlation between these two regions, meaning that when the medial prefrontal cortex activity is high, the amygdala is low. Okay, the amygdala activity is low. This is what we see in the adult brain, okay? And so it's interpreted as a reflection that the medial prefrontal cortex is regulating the amygdala in healthy adulthood, okay? There is top-down flow of information from the medial prefrontal cortex to the amygdala. And then, unsurprisingly, adults who typically exhibit this pattern, that is like anti-correlation, when medial prefrontal cortex takes over and regulates the amygdala, these are the adults that have better emotion regulation in real life, okay? Because obviously there is variability, not everyone is going to show this anti-correlation. And interestingly, we see that those that show this pattern have, so is really associated with healthy mental health, okay? So on average, okay, children don't show this adult-like pattern. They show a specific pattern, okay? They do not show this. So this child-specific pattern is what we use to put forward the argument that there is a sensitive period in the development of these connections between the amygdala and the medial prefrontal cortex. Okay? So this is very hard to study in humans, so we don't know for sure. But we had studied extensively this in rodents, okay? And so that's how we can inform the human brain research, okay? So in rodents, the period between weaning and puberty is the sensitive period for the development of the medial prefrontal cortex amygdala connections, okay? Between weaning and puberty in rodents, okay? They have studied this very precisely. Is my camera off? I don't think so. Okay, here I am. I just couldn't see myself. So they study this very precisely with something called optogenetics and tracing, and this is like it allows you to look at causation, okay? And that they have shown in rodents that the amygdala to medial frontal cortex connections develop earlier than regulatory connections in the opposite direction. So meaning first, what they see is the connections developing from amygdala to medial prefrontal cortex. So bottom up, that's developed first during the sensitive period. And then later on, you see the opposite. So consequently, the neurobiology of emotion regulation is qualitatively different in infancy compared to adolescence. So what they found is that the adolescent rat uses the medial prefrontal cortex for regulating emotion, whereas the infant rat, the pups, do not, okay? And in humans, we don't know the level of causation or anything like that, but we have seen that the amygdala and the medial prefrontal cortex connection continues to change from childhood through adolescence, mirroring what we see in rodents, okay? So what does this mean then, okay? What does this mean? What is the ecology of the developing child? This means that because the connections are still developing and there is only one-way communication from bottom up, it seems, and they cannot do top-down communication, they need someone else to do it for them, okay? The child needs somebody else to do it for them. So the caregiver serves as the external social regulator at a time of medial prefrontal cortex immaturity, okay? So parental presence is essential during early development. They provide the scaffolding for children's exploration and learning and emotion regulation. For an animal dependent on an adult for survival, there is a species expectation that the attachment figure will be available, okay? And that the caregiver serves as an external regulator at a time of medial prefrontal cortex immaturity. Again, this is why I don't support leaving babies to cry at night, because this is violating this very principle of brain development, which has mountains and upon mountains of evidence from rodent and from human research. So whenever I hear, oh, there's no sleep training studies that show this, why do we need to? We already know, okay? We don't need to look directly at this adverse childhood experience, which I consider to be an adverse childhood experience. Like as I said, parents modulate early emotional learning, and children learning to prefer and attach to parental cues, okay? So parental stimuli influence the connectivity, okay, of the amygdala and medial prefrontal cortex. So what do we have to do? Well, we need to provide the child with sensitivity, attunement, and co-regulation to build the infant brain, right? So, for example, let's look at each one. Let's look at sensitivity, attunement, and co-regulation. So, sensitivity, what is it? What is it really? Okay, well, let's look at how they measure. So in studies, really what they look at is how sensitive the mother is to a baby's cues, how much does she accept the baby's behavior. I say mother because all the studies are done on mothers, unlike parental sensitivity or the majority. Cooperation, how the mother cooperates with her baby, and emotional accessibility. All of those things are looked. Okay. And sorry, I just don't want to miss anything here. And so parental care, and then now going back to what I was saying about the connection between the amygdala and medial prefrontal cortex, they have found that attachment styles, okay, because these are all elements of attachment, sensitivity, attunement, and co-regulation, okay? Attachment styles in adulthood predict amygdala function. This has been shown too. So for example, studies have shown that attachment styles that are measured in adulthood are correlated with concurrent amygdala function. And when attachment is measured in infancy, these classifications then predict amygdala structural differences in 10-year-old children, okay? So attachment is very like the umbrella term, and then you look at all these things, sensitivity, attunement, and co-regulation. Really secure attachment means that all of these things are good and in the relationship. And then, interestingly, parental sensitivity during infancy is linked to smaller amygdala volumes, okay? While sensitivity during childhood moderates age-related increases in connectivity, with the suggestion that low sensitivity accelerates the development of functional connections, okay? So scientists seeing this as a bad thing. If there is an accelerated development of these connections, if they happen before the baby is ready, this is considered to be maladaptive. And then this is when later on we can see mental health issues. That is the hypothesis, have not been studied or shown in humans directly, but in rats, plenty, okay? In rats, plenty. So sensitive parents and the associated security established with the baby influence the nature of the amygdala, medial prefrontal cortex circuitry function in maturity, okay? Yeah, and so we all need to provide, or if we support parents, we all need to support the development of attunement, the development of sensitivity, and encourage them to provide co-regulation, okay? And if you're a parent, then you try to do these things. So attunement is when one feels felt, known, and accepted, right? You feel seen, really, and this is very good for you as you are growing up. You feel seen by your major attachment figures. And then co-regulation is like a back and forth exchange with the infant in a way that our response is based on the infant's response, and then vice versa, where we regulate the infant when they are dysregulated. So experience during the sensitive period of early childhood organizes brain systems. And one example that is very specific that has been shown in these two papers that I'm sharing with you is the amygdala and medial prefrontal cortex connections, right? Medial prefrontal cortex to amygdala connections. And so we can say that adverse caregiving experience are potent stressors for the central nervous system occurring during the sensitive periods. And studies in non-human animals demonstrates the adverse, as I said earlier. Their studies in non-human animals demonstrates that adverse caregiving accelerates, okay, amygdala development and promotes earlier growth. Presumably, this happens so that I can't see my screen anymore. So this is the explanation that they find. So they put rodents through like lots of stressors and like bad maternal care, and they see accelerated amygdala development. So it is hypothesized that this happens because the rat cannot rely on the caregiver, they had to mature fast. Okay? And so that's what's happened. But then when they're adults, they have problems with the stress system, okay? And so in humans, early psychosocial adversity may lead to accelerated amygdala medial prefrontal cortex development, but future research is needed, okay? We have more than enough evidence, though, to want to be cautious, right? To always operate on the cautious side. Here it is. So this is why optimal experience can set a strong foundation for later mental health. I hope I made my case with these two little studies, or they're reviews really, but they're very short and easy to read. So I encourage you to read. I'm gonna put the links in the description of the video. And I hope you enjoy this presentation. If you cannot get enough on what I'm saying, I encourage you to sign up for my ebook that talks about parental brain core concepts. Even though today I talk about brain development, the other side of the picture that really lets you to deeply understand what's going on is the parental brain science, which is absolutely fascinating. Or if you have more time, and if you're a professional particularly, I encourage you to watch my masterclass, the Parental Brain 101. If you have watched it already, you can only watch it once, or my system is going to kick you out. But if you want more resources, just talk to me, because I always can provide you in the right direction if you're thirsty for knowledge for this type of information. So I'm going to put all of these links in the description along with the studies too. Okay, so I saw that I took about 20 minutes, so this is a good length for a mini class. I hope you enjoy it, and I will hope to see you again in a month.